Those of you who heard the previous tape that we made in here will think you know this song. You're wrong. Little boxes, little boxes, little boxes in the genome. Little boxes, little boxes, little boxes all the same. There's a cat box, there's a tatter box, there's a Homea box, and there's a Pribner box, and they're all made out of ATGC. They all look just the same. So the scientists all clone them and run gels to read the sequences, and they all find little boxes all the same. And you might think that these boxes are boring with just 4 nucleotides. But remember little phosphates, 'cause they all look just the same. Little phosphates, little phosphates, little phosphates on the proteins, little phosphates, little phosphates, little phosphates all the same. They're on serine, they're on threonine, they're on tyrosine, and they're on histidine, and they form part of every nucleotide, and they all look just the same. And for every little phosphate, there's a kinase and there's a phosphatase, and they all try to move the phosphates so they won't all look the same. So the scientists all map them and use analogues to trap them. Then they all publish little motifs, and they all look just the same. Then the scientists read the papers and all rush to find the kinases, and they find one, then they find one, then they find one yet again. Then they name them and they blame them for life, death, and almost everything. Then they all publish little cascades, and they all look just the same. Now map kinase, 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 phosphorylates map kinase, kinase, and that phosphorylates map kinase, and that's where the trouble starts. 'Cause for every little kinase, there's a whole zoo of protein phosphatases, and they all take away the phosphates, so we're right back at the start. Then receptors in the membrane start the whole kinase cascade signaling. Then they all modulate the signals, little signals all the same. And the signals reach the nucleus and phosphorylate transcription factors, and they all bind to little boxes just the same. Little boxes, little boxes, little boxes in the genome. Little boxes, little boxes, little boxes all the same. There's a cat box, there's a tatter box, there's a Homea box, and there's a Pribner box, and they're all made of blasted phosphate, and they all look just the same. You'll all realize at once that the contents of this next song are so far-fetched that they couldn't possibly be true. Sadly, they are. It's a song that's dedicated to a noble British institution called British Rail, and the first 3 verses consist entirely of excuses for why the train network was seriously disrupted. And these were the actual excuses that British Rail offered. The remainder of the song consists of 2 true stories that happened to me while travelling from Cambridge. So this is a token of my esteem to British Rail. Dear passenger, there's something you should know. That when it snowed last night, it snowed the wrong kind of snow. While travelling and with this sprinkling on the ground, our high-tech wheels will not go round. They'll slide and they'll slip and they'll get no grip, and so our trains won't go. It's the wrong kind of ticket, it's the wrong kind of snow. There's something the wrong kind of temperature, that's why our trains won't go. That when it snowed last night, now the gentle breeze that disturbed the trees brought the leaves all a-tumbling down, and they all fell back across the track, all slippery and brown. And with these leaves upon the ground, our high-tech wheels will not go round. They'll slide and they'll slip and they'll get no grip, and so our trains won't go. It's the wrong kind of ticket, it's the wrong kind of snow, the wrong kind of temperature, that's why our trains won't go. Dear passenger, we've had a dreadful fright. The temperature dropped rather low, it even froze last night. And with their mechanisms froze, the doors won't open and won't close, so our reputation grows. 3 cheers for British Snail. It's the wrong kind of ticket, it's the wrong kind of snow, the wrong kind of temperature, that's why our trains won't go. Now I journeyed off to Kent one day for a meeting that was there, and I bought a return ticket, paying twice the single fare. And on the return journey, 2 inspectors joined the train. They said this ticket isn't valid, so you'll have to pay again. So I proved to the inspectors that I'd paid the whole full fare. They said then you can pay again, and they didn't seem to care. They threatened me with prison, they grew angry and confused. When their invitation to pay twice I steadfastly refused. It's the wrong kind of ticket, it's the wrong kind of snow, the wrong kind of temperature, that's why our train won't go. So they stood and argued half an hour, till a compromise I found. I proposed we paid each other the full fare of £20. So I paid them and they paid me, and with smiles all round they let me be. Have you gone mad or is it me? Please answer, British Rail. It's the wrong kind of ticket, it's the wrong kind of train, and one of us, I fear, has got the wrong kind of brain. But there's one time I must admit you excelled yourselves for sure. You replaced a broken engine in a quarter of an hour. When I wondered what had caused this speed and had caused you to react, I found the Queen was in the royal carriage at the back. She had the right kind of ticket and the right kind of rain, so I'll ask if she can come with me next time I take the train. This song is intended to make you laugh, but it may also make you think. Our attitudes to animals are curious and bizarre. As vegetarians keep dogs and give them flesh to gnaw, and our views on pain and cruelty aren't easy to define. We'd rather keep a killer cat than a gentle porcupine. So can we find a role model whose morals never stray, a paragon of virtue who can show us all the way, who does not kill or maim his food or other beasts offend? If you listen to me carefully, here's what I recommend. Just love that cuddly vulture, though the prospect may appall. Yes, love that cuddly vulture, it's a lesson to us all. If vultures don't appeal to you, would flies and maggots do? Their lifestyle's just as virtuous, they outshine me and you. They wait until their food has died and then begin to chew. So love that wriggly maggot, it's a lesson to us too. love that wriggly maggot, though the prospect may appall. Yes, love that wriggly maggot, it's a lesson to us all. As we evolved from animals with similar DNA, then should we show them more respect and not use them as prey? The problem with this concept is that plants are like us too, and if we can't eat them at all, then what are we to do? So love that juicy strawberry, though the prospect may delight, and remember to apologize before you take a bite. But another problem comes to mind if DNA's the key. Bacteria and viruses are just like you and me. The concept is quite simple, the message can be blunt. It's time that we all join the Smallpox Liberation Front. So love that deadly virus, though the prospect may appall. On second thoughts, let's leave it there, I can't love that at all. But one more snag occurs to me, if vultures are the key, and if we have to follow them, it's stiffs for lunch and tea. We'd have to eat our ancestors and gnaw their limbs with glee. For Hannibal Lecter, that's okay, but it's lettuce leaves for me. So love that cuddly vulture, though the prospect may appall. Yes, love that cuddly vulture, it's a lesson to us all. This song came from March this year, but it describes an incident which happened even longer ago, namely in the spring of 1965. And in case people ask afterwards, yes, it is based on a true story. Once I was a folk singer, I played a large guitar. I rolled around the folk clubs in an old and rusty car. I sang to lots of people and I made a lot of friends. And I wondered if my life could ever be so good again. 'Cause the joys of singing brought a thrill I can't explain, as the power of music feels so strong. Yes, the joys of singing brought me happiness again, and the memories of that time still linger on. I sang in 60s folk clubs, one I loved was on a farm, where Saturdays 400 people packed inside a barn. And when I sang the gentle notes of a love song sweet and slow, the calves inside the pen would answer, mooing soft and low. And then someone invited me to sing a double bill with a singer from America.
Welcome everybody to the second day of our cell biology practical course. And at the very beginning, before I'll go to the content of today's presentation, I would like to explain once again to all of you that these questions which in between I ask you and expect the answer in our chat room are not part of the exam, but they are just to stress the fact that we are live online and that we have a possibility to communicate, to ask about things which are not clear enough, and to motivate you to follow the text which I have prepared in the presentation. So along these lines, I would like to ask you right away a short, brief question, 2 points. Please write in the chat, what do you think, what is the charge of DNA and what is the charge of the plasma membrane of a cell? And now I will come to the topic. So as we told you yesterday, the first experiment is called immunofluorescence. The goal is to detect certain organelles within the cell. And in order to be able to identify them easily, we will use a technique of overexpression of marker proteins which are specific, typical for certain organelles. And for this purpose, we need to use a technique which is called transfection. You have heard about the transfection already in my cell culture course. You have seen a huge list of different possibilities, different techniques which are available, and also principles of these techniques. What we have chosen for our practical course here is the so-called calcium phosphate transfection. This technique was first published in 1973, so it's a pretty old technique. The reason why we have decided to use this one is that it's very popular, it's easy to be done, it's inexpensive, which is also a fact which is important for a large group of students. And it's relatively easy to learn, as you will see during my today's presentation. In the past, it was used for many different types of cultured cells, and it is used for both transient and stable transfection. And so we get already first answers. Some of you have written that DNA is negatively charged, which is correct. And another one, everybody says DNA is negative, that's correct. And the second part of the question was, what is the charge of plasma membrane? So inside the cell is negative, the outside is positive. We have to wait for more answers in order to come to the correct solution. How would one do the transfection? So as you can see, here is written that it is good for transient and stable transfection and for different types of cells. Every coin has two sides, and the problem of this technique is that it's prone to variability. It's sensitive to pH, temperature, and buffer salt concentration. It can be toxic, means that before you start applying this technique for the new cell type, you have to test it. It's not really efficient in primary cells. And the transfection efficiency, that means how many cells from the whole culture are effectively transfected, is very low. So that means it's below 1%. And superior to this technique is so-called lipid-mediated transfection with commercial reagents like, for instance, lipofectamine. However, these techniques are very expensive, so that's the reason why we cannot use them in the practical course. The poor transfection efficiency is a disadvantage, but in our case it's an advantage. Why? Because in immunofluorescence you need to distinguish between positively transfected and not transfected cells. And if you don't reach very high efficiency, then not all cells which you see on a small space with the microscope are positively transfected. That means that within the same sample you have also the, so to say, negative control. That's the advantage of this situation. The principle of calcium phosphate is co-precipitation of DNA with calcium chloride and with buffered saline phosphate solution, which generates a co-precipitate. So the answer to my questions was correctly answered by, for instance, Elias Wieser, saying that both are negatively charged. This is correct. So we have a negatively charged plasma membrane and we have a negatively charged DNA. So how we can then succeed in the interaction? The only possibility is to mix it with something what would neutralize the negative charge of DNA. In our case, it's the calcium chloride. So calcium chloride combined with HBS, this buffer will change the charge, and then this precipitate will not be negatively charged anymore, and therefore will have the ability to interact with the negatively charged plasma membrane. And then the next step of this procedure is that once this co-precipitate is on the surface of cells, it will be taken up by the process of endocytosis. On the bottom of this slide, you see the 4 steps of the whole procedure. So the first one is mixing DNA with the transfection reagent. In our case, it's the calcium phosphate. Then the next is the formation of this precipitate, which is the step 2, is the incubation. The next step is the adding of this precipitate to the cells. And the next step is further cultivation of cells. This depends on the type of cells, on how fast they divide, and on the goal of your experiment. If you want to see cells which are transfected but have little side effects of this transfection visible, so then it's probably better to cultivate them longer, so the efficiency will be lower, but on the other hand you will not see artificial effects of transfection. Or you need high yield, and then you don't need to wait so long because you need to have as many cells transfected as possible. So now going to the next slide, this is the protocol for the transfection of HeLa cells. As I told you, we are going to use the calcium phosphate precipitation technique. That means that in the first step you have to prepare cells. This is what I have shown you yesterday. Seed cells into a 6-well culture plate in 3 milliliters of medium and so on. 24 hours later, on the day of transfection, the cell density should reach 60 to 80% confluence because the transfection efficiency is sensitive to cell density. So that was the reason why we have counted cells in the first experiment. That was yesterday, because we knew that was tested already how many cells we need per well in order to reach this confluency 24 hours later. And then we come to the particular experiment, which is today's experiment. So we want to have a control which are not transfected HeLa cells, and then we transfect the second well with the plasmid construct DNA, which is coding for RAP5 small GTPase, which is GFP linked, and this GFP is there because we want to detect the localization of RAP5, and this is the easiest technique where you can directly see the fluorescence emitted by GFP. The next one is RAP7. What is the difference between these two? Why we have decided to use these two constructs is that RAP5 is the marker protein for the early endosomes, and RAP7 is the marker protein for late endosomes, lysosomes. So this is the reason why you have to compare in this experiment not transfected cells with cells transfected with RAP5 GFP or with RAP7 GFP. Then to the protocol itself, to this calcium phosphate transfection. So what you need for that, you will all find on your protocol book site page number 6. And what are the components which we need? So this is this HePES buffered saline solution, calcium chloride solution. These are the cheap reagents which one can prepare himself. Important is that they have the proper pH that has to be very carefully checked. These solutions are prepared in advance and in aliquots frozen at -20. However, before the experiment you have to pre-warm them to room temperature. For each transfection you have to prepare 2 tubes, so label tube A and B. In tube labeled A is always this HePES buffered saline. This is the same for all transfections. It's always the same volume. For this particular experiment we use 200 microliters. And then for solution B is a diluted mixture of your particular DNA, which will be mixed with calcium chloride. Why calcium chloride? Because calcium has a positive charge, is 2+. So with this one you can affect the negative charge of your DNA, and at the end you will form the complex which can interact with the negatively charged plasma membrane. Now we have in this particular experiment 3 different samples. This is just an example. So what we decided to do was to test different amounts of DNA, always the same DNA. So this is RAP7, either 1 microgram or 0.5 micrograms or 0.25 micrograms, and always the same volume of this transfection reagent. Why we do that? Because the efficiency of transfection always depends on the ratio between your DNA and the transfection reagent. So we wanted to do also an experiment where we will compare different amounts of DNA with the same amount, same volume of transfection reagent. So this is just the explanation why do you have it in protocol written like this. So what you have to do, you have to pre-pipette water, calcium chloride, and add the DNA, then properly mix it. But properly mix it means every time the same way, because you want to have identical conditions, and then incubate it for 5 minutes, incubate at room temperature. And then once this complex is built, complex means that you have to mix this solution B with solution A. I will show you everything in the film which we have prepared, how you do it, how you prepare it. Then you add this directly to the cells. So this is an advantage when you compare this technique, this calcium phosphate transfection, with, for instance, lipofectamine transfection, where you have to change the medium, because as I told you yesterday, we use the so-called full medium, which contains also FCS and penstrep, and these antibiotics have some negative effect on the transfection efficiency with the lipofectamine, so you have to change the medium for medium without these 2 components. In our case, in this experiment, what we will do here, we don't need to do that, so we just add directly to the cells, but slowly and dropwise, because we want to get a chance to this mixture to get in touch with all cells on the plate. So that's the reason why you should not rotate it, and you will see that in the film how to do it properly, and then incubate it at 37 degrees in the incubator for at least 5 hours. After these 5 hours, you change the medium, you add fresh medium and incubate the cells at additional 24 hours. Why you have to change the medium? The reason is that in certain situations, this such mixture for this transfection may be toxic for the cells, and if you want to avoid toxicity, you add fresh medium, so you replace the medium with fresh. Plus, we have additional experiment. As you have heard yesterday in the theoretical part, we are going to treat cells with Taxol, and you will see the effects of Taxol on the growth, on proliferation of HeLa cells. And in this step, we will also add this Taxol and leave it overnight in the incubators with the cells. So now we are coming to the first movie, and here is part of your protocol book where you can see that the solution A is always the same, but solution B, we have on one hand this experiment where we want to transfect RAP5, and then we have RAP7, different concentrations, different amounts of this DNA, always the same amount of the transfection reagent. However, before you will start the experiment, you have to calculate, this is one of your tasks, to calculate the appropriate volume of DNA. Here you can see in the protocol is written everything in micrograms, and we have stock solutions which are highly concentrated DNA stock solutions, so you have to calculate how many microliters of each you need in order to reach these amounts for the pipetting and so on. I have one more question. So the first one you have answered correctly, but in between, when I will show you the movie, you can think about the difference between the transient and stable transfection. The experiment which I will show you here today is the so-called transient transfection, but what I wanted to hear from you, and you have seen that in this cell culture series of lectures is what is the stable transfection. So how one can achieve this stable transfection. You can simply write very, very short answer. So we can start with the movie, and here you see that you have to calculate how much of DNA we are going to use, how much water we have to add, and then we start here. So this is first for the solution B, the biggest volume, which is water. Because the DNA is highly concentrated, water is sterile, double distilled water. The only part of this whole components which is not sterile is the DNA, because we cannot autoclave it, we cannot do anything with that, but the only danger is that there can be some toxins from the E. coli, but we are using Cryogen Maxi Preps kit where it is cleaned, so that's not a problem. To the water, you add the 24 microliters of calcium chloride, so that's the second component. You have to be careful by pipetting and collect everything on the bottom of the Eppendorf tube. And then the third part of this mixture is the DNA itself. So in this case, for instance, 1 microgram of this RAP7 GFP corresponds to the 6 microliters. So the last one which we add is the DNA. DNA has to be properly mixed, and then you add it slowly. You have to be careful because it's very small volume, not to have more from the outside of the tip than inside. Properly mix it, and after that you have to flick it with your finger in order to have a homogeneous mixture. And then we get to the next step, and this is adding solution B to solution A. That means that we will add this mixture of the DNA with calcium chloride in water to the HePES buffered saline. Important is to do it in this direction and not the opposite way, so always add DNA to this HePES buffered saline. First you have to mix once again, because it was incubated for 5 minutes, this DNA mixture, and then add it dropwise. Always flick the Eppendorf tube. This is the advantage of the movie, because you cannot normally see these small details when somebody shows you the protocol, but here you can see nicely that it will be mixed. What can happen is the solution may become turbid, milky, and this is exactly what we want to achieve. So this is an advantage if you see that there is a precipitate formed, because you see that the reaction runs and you were successful. It may be visible, it must not. It may also happen that it's not visible. But in any case, you have to do it always the same way. So mix it 15 times, then leave it for 15 minutes. Always the same procedure, because otherwise it's not reproducible. And then you add this mixture to cells, once again dropwise, and spread it all around the well. So here you have to be careful. We are getting answers on the previous question, and I will just finish this movie and then we'll have a look on your answers. So now you add dropwise this transfection mixture to the cells. You see that I put it into the center well and onto the left one, because the left one are the not transfected cells. You spread it evenly on the whole surface, so all cells will get the chance to get transfected. And once this is finished, we have to incubate cells in the incubator for at least 5 hours, and then the medium will be changed. So you see that it's evenly spread. You have to label your 6-well dish in order to know what was transfected where. Naturally, you have to put there all data about your cells, your initials, date, and so on, and then you start with the incubation. So why there may be a problem with the transfection efficiency. So one problem may be, and that's also a reason why we are not using lipofectamine transfection, is a so-called autofluorescence. We don't want to have artifacts, shiny points within the cells, and that's the reason why we are using calcium phosphate, because this has less such effects. The other option how to avoid this problem is that you keep cells in the culture, you cultivate them for 24 hours, and then you split them once again. So the disadvantage is that you will get less positively transfected cells. On the other hand, you will have less artificial autofluorescence. So now we have here some answers. Stable transfection is when you introduce DNA into cells long term, so the cells pass this DNA on their progeny. This is correct, this is the goal, but how was the question? It's mainly for protein production. In transient transfection, you look for gene expression changes. May be true. With transient transfection, the DNA which was introduced will disappear after a few generations also. But the point, the reason why I asked you was what we have to do in order to achieve the stable transfection. So we have to do so-called selection. We have to use antibiotics. And if we have in the construct antibiotics resistance, then you can select only those cells which were positively transfected, and you proceed with using these cells, and it will be in the future ones also passed to the progeny. So that's the way, that's the difference, which we are not using this step in our today's experiment. Now, after these 24 hours, we have to split HeLa cells to cover slips. Why? Because you need to do the immunofluorescence, and that's not possible to do on this plastic dish which you have seen before. So we have to make a small trick. We will bring them on cover slips, on glass cover slips. Secondly, we will also do the treatment. So what you have to do in this part of the protocol is the aspiration of the medium. Then all steps as you have seen yesterday, how we split cells. So you have to warm to wash them with warm PBS. Why we do that? You know that you don't want to inhibit trypsin. Then you add small volume of trypsin, so these washing steps are not important, the volumes. Trypsin only that much that you can cover the surface of cells. Then you incubate it, and then you stop the trypsin activity by addition of the full medium with the FCS. Afterwards, you collect cells. You have to be careful because now you don't have millions of cells. There are much less cells, and not all of them were positively transfected, successfully transfected. You put them into the tube, centrifuge in order to get rid of this trypsin. And in the meantime, you prepare a 24-well plate with 6 cover slips, which are shown here. Untransfected cells come into wells 1, 2, 3. RAP5 GFP transfected into this well, and RAP7 into these 2 last wells. You have to label it properly, because otherwise you will not be able to identify your samples. And after this, you add medium to each well. It has to be the same volume, so what we need for these smaller wells is only 1 milliliter. Then, once these cells were centrifuged, you get rid of the whole medium with trypsin by aspiration. You resuspend them always in the same volume with the full medium, and pipette only a small volume into these wells of the 24-well dish. After 5 to 8 hours, these cells have to be adhered to the glass plates, and then there will be added Taxol. So the next question was, not today, but in principle in the experiment when you do it yourself, how much Taxol you have to add. So we have a stock solution which is very small volume of 1 millimolar Taxol, and the final concentration, the working concentration, is 10 nanomolar. So one task which you have to do is to calculate the dilution, how much you have to add, and this you will add then to the well which is labeled A3. Cells will be incubated overnight in the incubator, and then we can proceed with the next part of the experiment. So you will see how to handle these cover slips here. So what we have to prepare is a 24-well dish. The same story as with the 6-well dish. On the upper left corner you have a mark, it's cut diagonally. Then you have to label it properly, because now you will have many more samples. So one part of it is not transfected, and then the rest is transfected, either with RAP5 GFP or RAP7 GFP. And then you will see how to work with the cover slips. So these are autoclaved, sterile glass cover slips. They have different sizes. Be careful when you start working with them. You have to be sure that you have the proper size which fits into the 24-well dish. They were autoclaved, so they are sterile. The unpleasant thing is that they stick to each other, and therefore you have to separate them with these special forceps. Forceps have to be also sterile, so we either flame them or clean them with ethanol, and then you put always one single cover slip into each well of the 24-well dish. So it's necessary to learn a little bit how to handle these cover slips. We had the advantage that this movie was cut and merged together, so even with shaky hands you can reach a successful stage with your prepared dish. And then you have to add everywhere the same volume of media. So it's the full medium with serum and with pen strap, with sterile tips. That's the DMEM. And this is the mixture of cells which were trypsinized, resuspended after decentrifugation, and we put everywhere the correct volume of cells, either control cells or transfected cells. Once again, it's necessary to resuspend them to have the same cell density and number of cells, and the idea is that they will grow on the top of these cover slips. So that was the reason why I put first the cover slips into the wells, and then we put the mixture of cells in the medium on the top of these cover slips. As in every experiment, after even spreading in the well, you have to check the situation in the microscope. So here you see trypsinized, rounded up cells. These are not dead cells, but they are freshly trypsinized. That's the reason why they are rounded up. And then you see also on the left side the edge of the cover slip. So here you see the difference between the glass on the right side, cells which are sitting on the top of this glass cover slip, and the plastic surface of the well on the left side. So that was the next part of the protocol. And that was basically the first part of this second day. The next will be the immunofluorescence procedure itself, where I will show you how these samples will be treated in order to detect in these cells transfected proteins, organelles, using various different techniques, which techniques you will see in 10 minutes. So we will start with the next part of our presentation in 10 minutes.
I'd like to take this opportunity of telling you what's been happening in the lab since I last addressed you. First slide, please. Now I'm sure you'll all remember what I said here last November, when I said what I had discovered with such ease. First slide, please. For the sake of time reduction, I'll miss out the introduction and go straight into the details. Next slide, please. Next slide, please. This shows NMR 3D of MPF and TBP. We PCR'd P53 and I peak cyclins A to Z, as you can see. Next slide, please. Can you hear me at the back? Well, can you hear me at the back? Oh, now the microphone's got feedback. Next slide, please. Next slide, please. Please, can someone shut that door or stop the rock group in next door? And please, can someone kindly catch that swarm of bees? Next slide, please. I'm sure you can't make out the tracks, 'cause this has just come in by fax. I'm still expecting several more, so next slide, please. Next slide, please. Now this curve that goes straight down shows stimulation upside down or just a crack the wrong way round. I'm not quite sure. Next slide, please. Can you go 13 slides back? Or is it 16? I lose track. And no, I meant the left projector. Next slide, please. Next slide, please. Now this is looking rather blurred. Is it a plane or just a bird? I should have skipped that 7th whiskey. Next slide, please. Next slide, please. This may cause some slight confusion, 'cause it shows cold nuclear fusion. Now it's melting the projector. Next slide, please. Next slide, please. Have I been drinking too much liquor, or is that smoke there getting thicker? Can you find a new projector? Next slide, please. Next slide, please. It looks as if I'll have to shout. The fire alarm is winning out. Can you just try to pay attention? Next slide, please. Next slide, please. I don't think this slide can be seen. That fireman's hat obscures the screen. Can you just wait until I've finished? Next slide, please. Next slide, please. Now I'm feeling rather manic, but the audience is panic. Won't disturb my concentration. Next slide, please. Next slide, please. Now this looks like apoptosis. Can you turn off those fire hoses? 'Cause the water level's rising. Next slide, please. Next slide, please. I'd like to thank the organizers, those 2 penny-pinching misers, who would only guarantee my one-way fare. Last slide, please. I see the fireman's going frantic, so I'd best swim the Atlantic in despair. This is a nightmare experienced by most scientists. As I was walking one summer's morning, I made my way to the library in search of wisdom and education. I scanned the journals so eagerly. I picked up Nature and looked inside it, and lots of adverts I there did see, for PCR and restriction enzymes, for cloning kids and HPLC. At last I tracked down the page of contents and scanned the titles so eagerly. There were quarks and quasars and plate tectonics, T-cell receptors and HIV. And as I browsed through the latest scandals, I sighed and yawned rather wearily, till there I saw my latest paper, and brightened up most immediately. I swelled with pride as I gazed upon it. What finer gels could you ever see? And every figure was as we'd planned it. Models of logic and clarity. But then I paused in deep contemplation. A nagging doubt stirred to trouble me. It was just 3 days since we'd sent the paper. There was no way this could be by me. The author's name and address were different. All other features were just the same. I gazed in horror at every figure. There was no doubt we were scooped again. I muttered oaths as I gazed upon it. I mopped the sweat from my fevered head. I'd have torn my hair, but that posed a problem. So I sighed and wished that I'd stayed in bed. I found a keyboard and started writing. A short erratum I sent straightway, correcting names and address of authors. And whistling softly I strolled away.
So welcome back after the small pause in this second part of our presentation of the cell biology practical course. So what I want to show you now is the next procedure planned in this experiment in the immunofluorescence detection, and that is the technique how you would prepare your samples for the immunofluorescence microscopy. So I will start once again with a question and try to write a very short, brief answer. What do you understand under the term indirect immunofluorescence? What is indirect immunofluorescence? The reason why I'm asking you that is that I'm going to show you different approaches how we can prepare these immunofluorescence samples. You will see that there is this indirect immunofluorescence and also something else. So what we need for this particular protocol, which we are going to use in our experiment, is the so-called cytoskeletal buffer, where is as one component pipes. These pipes is buffer, and then salt, EGTA, glucose, magnesium chloride, and so on. So this was tested before, it's optimal for this technique. Then one important part of the experiment is the fixation, where we use PFA. This PFA stands for paraformaldehyde. Usually there is a stock solution which is frozen at -20, which is 16%, and out of this one prepares the working solution, which is 4% in this cytoskeletal buffer. And this can be aliquoted and stored for a short time at 4 degrees. What is necessary to know is that this paraformaldehyde is toxic. Therefore, I will go back to the first slide where is some general information, and that says that be careful, paraformaldehyde is toxic, and you will see in the film that we work in this laminar flow hood. So this is not exactly the same one which is used for the sterile work in the tissue culture room, but it's also laminar flow chemical hood which is protecting the person working with such solutions. And the second point is you should not drop this waste into the garbage or sewage. You have to collect it and dispose separately. So this is a toxic chemical. How does it work? It's causing covalent cross-linking of, for instance, proteins with DNA and so on. So the structures which will be protected as they are in living cells. That's the advantage of this system. Next one is dye, which is usually in laboratory slang called Höchst. There is a number, there are many different types, but basically what we understand under this Höchst is a dye which is binding to DNA. It's not intercalating in the DNA, but it's binding to the minor groove of DNA and allows us to counterstain cells in order to localize the nucleus. However, it's necessary to know that it interferes with DNA replication during the cell division, and therefore is mutagenic, carcinogenic. It has to be very careful. You have to be careful when you are handling this reagent. Use always gloves, and we dispose solutions. It has to be also separately collected. All steps of this immunofluorescence procedure are performed at room temperature, except for the very first step, where you will see that I have PBS, this phosphate buffered cell line, on ice. And what is very important, what you have to learn when you start working with immunofluorescence protocols, never let cells dry out. Why? Because this will increase the background, so it will be milky. There will be no contrast. You will not be able to identify the localization of your proteins, organelles of interest. These cells in our particular experiment were transfected with GFP, with the fluorescence tagged protein, which was RAB5 or RAB7, and therefore you have to be careful during the whole procedure not to keep them under strong light. You have to cover them always with aluminum foil because you want to avoid bleaching of the sample. So next solutions which you have to prepare before you start is a so-called Modul solution. This Modul solution you will see at the very end of the procedure is used to embed your samples. So it's also an anti-bleach reagent, anti-fade mounting medium, which is a so-called artificial resin. So this Modul we have also in aliquots. It's quite viscous. You have to be careful when you work with it because it's in glycerol, and therefore it's not so easy to pipette. Next solution which you need, and you will see in the protocol, is the so-called washing and quenching buffer. So what means quenching? Quenching is in order to get rid of aldehyde groups from this paraformaldehyde, you have to quench them with ammonium chloride in the cytoskeletal buffer, which is this first one. And then we have so-called blocking and permeabilization solution. And here we are coming to the question number 1 today in this part of the presentation. I ask you, what do you understand under indirect immunofluorescence? And one answer here is an unlabeled antibody binds to the target molecule. and second antibody, which is fluorescent, binds to the first one. And then we have also other answers from Elena Dušek, for instance, or Magdala Brandner. With indirect immunofluorescence, you use 2 antibodies. Yes, this is correct. So you know that we are going to use antibodies. And how would you get these antibodies into the cell? So you need to make some pores, holes, to say, in the plasma membrane. And that's the reason why we are using permeabilization solution. So in this permeabilization solution, you have these components which are listed here. Among them is so-called saponin. And this saponin is kind of detergent. Actually, it's a glycoside surfactant. So surfactants are something like soap molecules which can make small bubbles. In this case, it interacts with lipid parts of the plasma membrane. In our case, cholesterol and phospholipids. So with this solution, you can make these small pores, and through these pores will get your primary antibody into the cell. Now, this I explained you the fixation. Important is to notice that it says 8 minutes, and take care of removal of all PBS solution and so on. Why? Because you want to have reproducible conditions. That means that the concentration has to be 4% and not less, not diluted with PBS. 8 minutes, because we have tested this with HeLa cells. It's not good if you do it much longer, because you would overfix these cells and then the efficiency of the labeling will be not so good. And of course, cells should never dry out. Then is a washing step, which is with this CB buffer 3 times 5 minutes. Here I would like to tell you also a small trick or advice. These volumes, minutes, and so on are not very important. You don't need to measure the time 5.00 and 1 milliliter and so on. But what is very important is to do it 3 times. So as often as possible to replace this media, washing media and so on is good, because then you get nice clean samples. Remove cytoskeletal buffer and add 400 microliters of blocking and permeabilization solution. So now comes the next question: what means blocking? We want to achieve a very specific binding of our primary antibody to the antigens. And we don't want to have non-specific binding. And for that reason, we use a blocking buffer. What is blocking buffer? There are different compositions, but what we have tested and used is, for instance, combination of different sera. Sera means parts of blood from animals where are raised the secondary antibodies. So you block binding antigens which are non-specific by using of these different sera. Or you can use BSA, so bovine serum albumin, for instance. Staining. The antibodies are diluted in blocking and permeabilization solution, so that's the same one as you have used before. Which antibodies now? So we have here 3 different components which we are going to use in our immunofluorescence detection. First is antibody anti-LAMP. This LAMP stands for a protein, is abbreviation of a protein which is a marker protein of late endosomes, lysosomes, and LAMP stands for lysosomal associated membrane protein number 1, typically resident on lysosomes. It's a glycoprotein, and the antibody which we want to use in order to identify the localization of these late endosomes, lysosomes, is a mouse monoclonal antibody against a LAMP, which will be diluted 1 to 100 and so on. These are the details of the manufacturer and so on. That means that this is used for this so-called indirect immunofluorescence. So we have a mouse monoclonal antibody. That means that we will need to use a secondary antibody anti-mouse. And this secondary antibody is then fluorescently labeled. And then the next antibody which we are going to use is anti-alpha tubulin, mouse monoclonal antibody. So why we want to use this one? is in order to show you microtubules, to identify microtubules in the cells. Why we want to see microtubules? Because as you heard, as you have seen in the protocol already, we are using Taxol, and Taxol is affecting microtubules. So we are going to detect microtubules using antibody against alpha-tubulin. It is once again mouse monoclonal antibody, so we have to use anti-mouse monoclonal antibody. And then we have another option, and this is usage of so-called phalloidin Alexa 488 conjugate. So what is this? Phalloidin is directly labeled with this fluorescent dye Alexa 488, which is a green one. And this phalloidin is interacting directly with F-actin. So it's not recognizing actin monomers, but it's mainly interacting with quaternary structure of F-actin filaments. And this is a direct immunofluorescence, because here you don't need to use any secondary antibody. This is not an antibody, but this phalloidin is directly interacting with the actin, and you will recognize it by this green staining of Alexa 488. Why I'm mentioning that? The reason is that if we have here green staining and we have 2 times the same species, so mouse monoclonal antibodies, and we are going to use an antibody against mouse monoclonal antibody, we have to be careful. So this can be, here is this green, this must be red. But if we have 2 times the same species, then we cannot use all these antibodies in one sample. And that is the reason why we have prepared several samples with the same transfection. So we will then combine always green and red staining, either by using LAMP antibody and secondary anti-mouse, which will be, for instance, red, and we have a GFP labeled protein which is green, or we have once again anti-tubulin, this mouse monoclonal tubulin antibody secondary, which is labeled red, and we have GFP, which is green, or we have Alexa 488 conjugated phalloidin, which is green, and this we can combine with one of these which are red. The reason why I'm mentioning that is you can once again write briefly just one single word. What happens if you have red and green staining at the same spot? What is the final color which you can see in the microscopy? What do you expect? So now to the protocol itself, we will have prepared mixtures of these primary antibodies or this phalloidin and primary antibody labeled as A, B, and C. So this is phalloidin, which we know already that it's labeled with Alexa 488, which is green. Anti-tubulin antibody, which is not labeled, so has no color, but we will use a secondary antibody against this mouse monoclonal anti-tubulin. Then we have a second sample, which is the solution anti-LAMP antibody alone. Then we have solution C, which is blocking and permeabilization solution only. So I see here are coming answers, yellow, yellow, yellow. So this is a correct answer, red and green is yellow. The reason why I have asked you about this combination is that we are going to use it for one particular purpose, and this is called colocalization. So if we expect that 2 different proteins are localized at the same organelle, and one of them we detect with antibody, which is then either green or red, and the other one is directly labeled, for instance, green. So then we know that if they colocalize these 2 proteins at the same spot, we have to get yellow staining, so we will have a yellow spot. If they do not colocalize, that means that, for instance, a marker is for late endosomes and the RAP5 localizes at early endosomes, then these red and green spots should be separated and will not give yellow staining. So that is the reason why we are using that. So going back to the protocol, what we will do, we will prepare a so-called wet chamber. So this sounds very scientifically, but in reality it's this simple Petri dish where you see several layers of paper which is soaked with PBS, so it's wet, and then a piece of parafilm which will be on the top of this wet paper. On this you have to label what is which position, position number 1, 2, 3, and so on. And there you apply approximately 30 microliters of antibody solution. So this is the solution A, B, or C, so these primary antibodies. And that has to be carefully done, so they should be separated well, because you are going to use, for instance, identical samples of cells with different combinations of antibodies, so you don't want to mix it together. So this is just the scheme how you prepare these 6 spots with these drops of your antibody solutions, and then you go to the next step. You put these cover slips with cells on respective antibody dilutions and incubate for at least 1 hour. How? I will show you, because this is a little bit tricky. You have, first of all, think about the position of your cells on the cover slip. They must be on the top of the cover slip, but if they have to interact with this antibody solution, then you have to turn it around. So upside down you put your cells on these drops. Then you close the whole Petri dish, and then you have to protect it from light. That means that you simply put it somewhere where there is no daily light and no artificial light and so on, because you don't want to bleach your GFP, and it will also interact with your antibodies. Then the next step is washing step. Once again, it's not important if it's 500 or 400 microliters, if it's 5 or 4 minutes, but important is to change as many times as possible the washing and quenching buffer. So with this washing buffer, you get rid of this whole antibody which was not specifically interacting with antigens. And you can do this either in this wet chamber, which is shown here. So you just simply put here fresh solution and suck it away with the vacuum pump, or you can remove the whole cover slip and put it into small 6-well plate with reasonable volumes of buffers. Important is to change this washing buffer as often as possible. Then you go to the next step, which is the secondary antibody. So the secondary antibody is recognizing primary. That means that it has to be against mouse, and it is also directly labeled with a fluorescent dye. So it's either Alexa 488, which would be green, or 586 and so on, which is the red. Or what you have in one case was a sample where is only blocking and permeabilization solution. So that means in that case you should not get any signal. You use the secondary antibody, but there was no primary antibody. That means that this sample has to be at the final stage when we are doing the microscopy completely black. So that shows you the specificity of your primary antibodies. Due to the blocking and so on, they interact only with specific antigens, and you will not see anything if you don't have any antibody, any primary, because the secondary will not recognize anything there. So this is to explain the protocol. You incubate samples for additional 45 minutes, once again in a dark space. Then you do the washing step many times, at least 6 times 5 minutes. And then at the very end, in the last washing step, you add this so-called Höchst dye or DAPI. There are different ways how one can detect DNA and counterstain nuclei. So you see that written here. Usually very high dilution is enough. So you can use either 1 to 5 or 1 to 10,000 dilution. And then you have nice strong staining, which is specific only for the nucleus. One more washing step, and then mounting of your samples. This mounting is, as I told you, into this Modul. So Modul is this artificial resin, and then you have to leave it polymerizing at least 1 hour. Usually we leave it overnight in the aluminum or dark aluminum foil covered space at +4 degrees, which means in the cold room. Now here is the movie, and you can see that protocol from the very beginning. So we are taking out living cells which were transfected, which were treated with Taxol, and we go to the microscope, because I told you it's always important to check at every step what happened with your cells. And what you see here, cell density is too high for immunofluorescence. How would you recognize that? First of all, you see that this is, for instance, a monolayer. So here you see the cell contacts, but in this area are several layers of cells, because it's HeLa cells. These grow upon each other. There are multi-layers. There are dying cells. These are the small roundish dots. It's probably also moving, and this is not ideal situation for immunofluorescence. So for immunofluorescence, we want to see individual cells. For instance, here we have a space where are no cells, and probably something like this would be ideal for our immunofluorescence image. You don't need to have too many, because we are doing large magnification. So everything what is moving are dead cells, and that's the reason why you have to do properly the washing. Then you have seen that this is the part where is the cover slip. So this is the glass, and this is plastic. On plastic the density is not so high, here is higher. But important is that they were growing well. Also on the glass cover slip there are cell types which are not growing properly on the glass without special coating, so you have to prepare a special glass coating. Now you aspirate the medium and wash cells with PBS++. So what means plus plus? That was this written on the bottle here. It's calcium magnesium. This is important to notice, because you have to prepare the solution before you start your immunofluorescence experiment. It is written in the protocol, the concentration and so on, because you want to fix cells as close to the natural conditions as possible. If you wouldn't have this calcium magnesium, then they would probably already start to open their cell-cell contacts, and they will not be in that shape as we wish to see them. These steps are not performed under aseptic conditions, so this is not done in the tissue culture hood. This is in normal lab. You can aspirate this PBS. You have to wash it as well as possible several times, and be careful, don't pipette directly on the cells, don't touch the cells with the tip, and also when aspirating this PBS, don't touch cells, go always to the edge of the well, but do it properly, because as I told you, the next step is the fixation, so you don't want to dilute PFA. Now we are going to this step, under the flow, fix cells with PFA, 4% solution, and as you see, this was done in the chemical hood. The volume is once again not so crucial, but important is to cover the whole surface. Do it as quickly as possible, they should not dry out. So you just simply add it on the wall, on the side of the well. And then it is important that you will not just leave it standing, but it has to be shaken. So it is in this rotary shaker. Don't do it brutally, because you will lose the cells. So gentle shaking is enough, but it has to be continuous, and you have to check the time, because as I told you, this depends on the cell type. In the meantime, you prepare the wet chamber. So this is the simple thing. It's a Petri dish with filter paper or paper towel soaked with PBS, parafilm, which is labeled, so you know the positions where is which sample. So this we call wet chamber. Then you prepare these dots of primary antibodies. So this is the primary antibody solution, which was centrifuged properly before. So at high speed, you want to get rid of all insoluble particles, because these particles would interfere with your immunofluorescence image. The distance between these samples has to be proper because you don't want to mix it together. So you prepare this. In this case, it's the same solution, but naturally in the protocol is written what is what. You can use different solutions of different antibodies. And then you take out your cover slips, which are fixed and washed. You have to get rid of unnecessary liquid. So you take the paper towel and you dry it from the side. Here, be very careful which side is with cells and which side is the bottom. And then you put it upside down, so cells towards the antibody solution on these drops of primary antibodies in the wet chamber. You can nicely see the cells even without microscope because the glass is milky and on one side is shiny, so that's the bottom of this cover slip. And don't forget that the cells have to face the antibody. I'm repeating it several times because our experience is that always somebody forgets this and then there is no result in the immunofluorescence experiment. Next is the incubation in the dark. After 1 hour, you wash cover slips. So there are different options how to do it. One option is just do it directly with pipetting and aspiration. The easier solution is to take your samples and put it into a 6-well dish. Why easier? Because here you have certainty that they will be nicely washed. They will be in a reasonable volume of the washing buffer, and you can put it on the rotary shaker for these 5 minutes 6 times, and then you are sure that you have nicely clean samples. So we put it on the rotary shaker and gently shake it for 5 minutes several times. Important is to do it gently because these cells are not so robust. You lose the sample. In the meantime, you can prepare slides where you have to label what is what on left and right side. You can put on one microscopic slide at 2 samples. Don't put them on the edge because that's not easy to do the microscopy. You cannot mount it on the table in the microscope. And after the secondary antibody and washing of the secondary antibody and staining of the nuclei, you have to mount it with Modul. So I told you that it's very viscous solution. That was the reason why I cut this tip. And then it doesn't matter what is the volume. It's approximately 30 microliters. But important is that you should not make any bubbles because that would destroy the image in your sample. You will not be able to do the microscopy. And then after secondary antibody, after washing, after staining of nuclei with Hoechst, after additional wash, you take the samples out. And now you should not forget to turn it with cells downwards, so upside down, because your cells are between the cover slip and the slide. So this is very, very important. Then you have to get rid of the unnecessary Modul. So usually I aspirate it with this vacuum pump. There should be no bubble because these bubbles will also destroy the image. And afterwards, don't forget to clean the vacuum pump because Modul is very viscous and it will clog it. Then you close it, protect it against the light, and put it either into the cold room or wait at least 2 hours until it's solidified. It's Modul, resin, and you can start with the microscopy. So now we are going to the expected results. First, I have prepared here a selection of fairly nice images from commercial sources or from publications. What you can see here, source is the manufacturer of the antibody. It's the company Epcam. And the point what I want to show you is the localization of RAP5. So on the upper left picture, you can see RAP5 in green. Why? What was used? Why is it green? So these are HeLa cells, the same ones as we have in our experiment. They were fixed with PFA. they were permeabilized with triton. So we have saponin, which is a little bit more gentle than triton. And then on the left side was used antibody. So in this case, it's not a GFP RAP5, but it is RAP5, the native one, detected with anti-RAP5 antibody. The secondary antibody was goat anti-rabbit. That means that this RAP5 was produced in rabbit, so it's a polyclonal antibody. This secondary antibody is labeled with Alexa 488, so this is a green dye. That's the reason why it's green. This image is a confocal image. That means that it is more sharp because it's only one confocal plane. It's very thin section, so to say. And what you can see, what you should notice here is that the labeling of RAP5, which is the marker protein of early endosomes, it's not only in the vicinity of the nucleus, but it's also in the periphery of the cell. So it's more towards the plasma membrane. These are these shiny dots here. And on the right side, you have a combination of green, which we have seen before. So this is the green with red staining. And this red staining comes from tubulin. It's an antibody against alpha tubulin. This antibody against alpha tubulin is recognizing tubulin, and tubulin is the marker for microtubules. So here you can nicely see these microtubules. These are these red stained filaments. And the blue staining is explained on the bottom, is the DAPI. So this is nuclear counterstain. In this case, it's called DAPI, this dye which they have used. We are using Höchst, but the effect is the same. So we can recognize where is the nucleus, and you can see also the shape of the nucleus, and you can also identify mitotic stages and so on. On the right side here is the so-called secondary antibody control. So this is the same as we do. As I told you, if you don't use primary antibody, use only blocking buffer, and then the secondary antibody as for all samples, the result is like this. If it's really specific, then you should not get any staining. So there is nothing red, nothing green, because this is only secondary antibody which did not find any primary antibody, so it's black. So this is the negative control. Next image is from a publication which is a recent one, and this is RAP5. In this case, it's eGFP, enhanced GFP construct labeled RAP5. So it's a protein which is expressing GFP there, where is RAP5 overexpressed. So it's not the same as the previous one, because the previous one was the native, and this is overexpressed, transfected in HeLa cells, so the same system as we have. And here you have, with these arrows, marked endosomes. So in this case, unfortunately, we don't have any counterstain for the nuclei, but you can imagine that this is at the periphery of the cell, so it's in the cytoplasm, and these larger dots here are the early endosomes, labeled with eGFP RAP5. So it's GFP, therefore it's green, and it's directly, so it's a protein which is labeled with GFP. Now, the next example is from the company Sigma. It's basically example of usage of an antibody which is recognizing RAP7. In this case, it's immunofluorescence in HeLa cells, so the same cell system as we have, fixed with 4% PFA and permeabilized with saponin, so it's exactly the same under the same conditions as we do in our experiment. And what you can see here is this RAP7 antibody, and secondary antibody is goat anti-mouse IgG conjugated to a dye which is 488, so it's green. And what you can see here is that these green dots are mainly in the vicinity of the nucleus. So RAP7 is a marker protein of so-called late endosomes. Therefore, it's not at the periphery as we have seen before with RAP5, but it's mainly here closer to the nucleus. Next example is also primary antibody against RAP7, but the difference is that these are not HeLa cells. These are HepG2 cells, which are human hepatocyte cells, which are tumor cells isolated originally from a 15-year-old patient with liver cancer. And what you see here are also cells fixed with formaldehyde, permeabilized with Triton, blocked with serum, incubated with this antibody against RAP7, and secondary antibody against mouse. So this is also mouse monoclonal antibody. What do we see here? Clearly, everywhere is the staining very close to the nucleus. So it's perinuclear staining, typical for RAP7 in the late endosomes, lysosomes. Next example is from a publication where HeLa cells were transfected with a similar construct as we have, GFP RAP7. And after 2 days, they were fixed, permeabilized, and then immunostained either for LAMP1, which I told you is a marker protein for late endosomes, or with an antibody against the protein called EEA1, which is a marker protein for early endosomes. And here you can see the result of this experiment. So they have overexpressed in HeLa cells RAP7 GFP, which is the green staining, very strong expression, and mainly in the close in the vicinity of the nucleus, which is unfortunately here not counterstained, but it is somewhere here, and here you can see the RAP7 staining. Then EEA, which is more in the periphery, these are these fine dots, these are the early endosomes, which are at the in the periphery of the cell. And then they used anti-LAMP antibody detecting the late endosomes, and you can see the similarity between the first and the last image, that they are fairly similar. That means that these two proteins probably co-localize, but in order to see the co-localization, you have to do the merge, and only those dots which would be yellow are confirming the co-localization. Now we are coming to the real results of an experiment which was performed by your predecessors last year. And here you see two examples, so from two different samples, where we have used the overexpression of either RAP7 with GFP or RAP5 GFP. How you can recognize which cell is positive, which was transfected successfully, so you have to compare this image with this image on the right side, which is the staining with Hoechst, detecting the nucleus, and here you can clearly see that we have only 2 cells which are GFP positive out of 3, so that means that this one was not transfected. The next image on the bottom, once again you see 3 different cells, only one of them is positive for RAP5. Now what we want to see are these fine dots which are typical for the overexpressed RAP7. So typical is not what is in the periphery but close to the nucleus, and you see the stronger staining around the nucleus, and that would localize to the late endosomes. In order to confirm that, we did the double staining. So the second is the antibody against LAMP1, which is the mouse monoclonal antibody. We use secondary against mouse monoclonal, which is red labeled, and then if you have the same localization of both, so you have red and green signal at the same space, and you make the merge, you can see yellow dots. So that's the colocalization, typically for late endosomes close to the nucleus. On the bottom is the LAMP1 once again, which is the marker for the late endosomes, but overexpression of GFP RAP5, and RAP5 is the marker for early endosomes, so it's more in the periphery. These are these shiny small dots far away from the nucleus. And if you compare these two images, this one with this one, you see that here on the top you have these yellow dots, and on the bottom you have either green or red, but no yellow staining. That means that here is no colocalization. And on the last image, which I want to present today, you see several examples of cells which were either not treated, so they were not transfected and they were not treated, HeLa cells, or you have same cells, but they were treated with Taxol. So you see the effects of Taxol when you compare it with these upper images. So what we do see here, you have to concentrate on the tubulin staining. So tubulin staining is for detection of microtubules, which are very nice. You see these flat cells, you see nuclei which are intact and have the right morphology. These are nicely dividing cells, and normal microtubules. Also here is an interesting image because you see a late telophase, which you can nicely appreciate on this part where you see these two nuclei, microtubules. So these are cells which were actively dividing now, and then effect of Taxol, which is blocking microtubules, and it's a stabilizing agent. So you see that the morphology of microtubules, mainly in this second image here in the center, is completely different, because here are these bundled, very thick microtubules, and you see that there is also an effect on the proliferation of these cells, on the mitosis. These nuclei are stained and the morphology is completely abnormal. And then you can also see cells, pre-mitotic cells with condensed chromosomes here, and also microtubule, so tubulin staining, which is not normal. It's not the same as you can nicely see here on these top images. So basically something like that we were expecting also from the experiments performed by you this year. And I hope that you were able to follow the whole procedure and understand the reasons why we do this protocol for the identification of organelles within cells. This is the end of my today's presentation, but we are staying still online for next 10 to 15 minutes. If you have any additional questions, please write it into the chat room or send us emails. Thank you very much for your attention.
Ja, super gut hast du das gemacht.
Danke vielmals.
Yeah. Corona 2020 recordings. I am the doctor. Corona 2020, ein richtiges Luder, aber macht die Rechnung nicht ohne Hess, Heiß und Huber. Online-Vorlesung geht ab, ziemlich nice. Um 11 Uhr stehen wir alle bereit, transportiert wird der Vibe ohne Spannungsgradienten. Huber, Hess und Heiß, die Unwissenheit wird enden. Sag mir, wer liefert Corona 2020 Recordings? Corona 2020, ein richtiges Luder, aber macht die Rechnung nicht ohne Hess, Heiß und Huber. Online-Vorlesung geht ab, ziemlich nice. Um 11 Uhr stehen wir alle bereit, transportiert wird der Vibe ohne Spannungsgradienten. Huber, Hess und Heiß, die Unwissenheit wird enden. Come on everybody! Sag mir, wer liefert Corona 2020 Recordings?
Es kann doch alles nicht sein, aber wir sind niemals allein.
Come on, everybody!
Wir sitzen alle da, sind am Ende mit unserem Batterie. Es kann doch alles nicht sein, aber wir sind niemals allein.
Come on, everybody!
Wir sitzen alle da, sind am Ende mit unserem Batterie. Es kann doch alles nicht sein, aber wir sind niemals allein.
Come on, everybody!
Wir sitzen alle da, sind am Ende mit unserem Batterie.